And when I say just around the corner, I have an address. (353rd Street). So 3rd Street is a street that's parallel or next to this building here, and it's just apparently up one block, turn right, and it's just on the corner. Okay, so I'm going to be giving the next presentation, and this is an opportunity really for those of you who are interested to find a little bit more about ProImmune and some of the services in the context of understanding and characterizing immunogenicity in more detail. So the title of the talk is Tools and Technologies for Comprehensive Immunogenicity Risk Management. It's probably quite a grand title, because I think anything that's comprehensive is always going to be maybe a little bit oversold, but I want to be clear that the reason we're calling it that is really just because we believe very strongly this is a very, very broad panel of assays that can be applied to questions of unwanted immune responses. So we've seen some similar slides to this already today, and in simple terms, what we're looking at here, of course, is the issues relating to biological molecules being taken up by the immune system, notably antigen-presenting cells. And in the course of this uptake of the molecule, the proteins are processed and presented in the context of MHC molecules. Typically this would be MHC class 2, but of course you could potentially also induce cross-presentation onto MHC class 1 as well. So peptides derived from these proteins are presented in the context of MHC and presented to T-cell receptors on the surface of CD4 positive helper T cells. And it's this specific interaction which is very, very important, because T-cell epitopes by themselves don't cause clinical immune responses or clinical immunogenicity, but they are required to drive that immune response. So following help, or this interaction between the MHC class 2 and the T-cell receptor, activation occurs and CD4 positive T-cell proliferation, cytokine production, and of course that the proliferation and division will mean there's a clonal expansion of these particular T-cells. Help is required to then drive these immature B cells that are specific for also the drug of interest to become mature, and this affinity maturation can only occur with the help of the T cells as well. So once the B cells become plasma cells, they produce antibodies, and these antibodies can then bind to your drug of interest. And of course that's not what we want, because that can cause reduction of efficacy, can alter our PK and PD readings, and also cause safety issues relating to cross-reactivity of self-antigens. So the whole of our sort of range of different assays that we provide at ProImmune is really geared to addressing different aspects of this, but particularly the key driver here relating to MHC peptide presentation and measurement of this presence of these CD4 positive T cells. So why are we interested in profiling epitopes relating to unwanted immune responses? There will be many different reasons that people in the room here are interested in that. One may be that you're in quite early stages of developing a biotherapeutic, and the question of lead ranking or lead selection may come to the fore. In this context, it's important to really understand what that total T-cell epitope content is of your biological molecule. That can be in a qualitative way, or it can be even trying to characterize the precise T-cell epitopes. Particularly in the context of novel scaffold proteins, for example, rather than just classical antibody molecules, it may be very important to characterize the details of T-cell epitopes in those new kinds of molecules. You may also want to evaluate the population impact of antigenicity. Many different diseases, particularly thinking about autoimmune diseases, will be associated with different HLA types. Rheumatoid arthritis being just one example. And in that particular situation, if you're developing a biotherapeutic, which turns out to have a relatively high T-cell epitope content with peptides that are actually restricted to the very HLA molecule which is associated with your disease, that can be a problem, of course, because it will disproportionately affect the patients that you're trying to treat. So detailed characterization of those epitopes can be very, very important. So maybe you've already selected your lead. What next? Well, maybe you're thinking in the stages of lead optimization. Then you may only have one molecule that you're working with in a particular area, and in this situation it may be important to identify particular regions that might be suitable for re-engineering. Re-engineering isn't something which people undertake lightly, of course, because the impact of introducing mutations can be quite drastic, relating to the potency of the molecule, but it may be only one option you have left with regards to reduction of immunogenicity. So this is certainly something which our clients are also coming to us and asking us questions about. You may want to identify specific epitopes in your biological molecule, not from a point of view of designing a molecule or selecting a different candidate, but actually to identify epitopes for then monitoring in patients. So more to do with the biomarker discovery and as a tool to then monitor the presence of these T-cell or even B-cell responses in patients receiving the therapeutics. So all this information can be really built together to support a strong regulatory position. Now the regulators in both Europe and in the US here have a very keen interest in trying to understand what these immune responses actually are and what affects them. But there are no obligations on molecule developers to do a particular type of assay in the context of preclinical immunogenicity. However, having said that, they are very keen to see that sort of data because it's informing them, particularly in the area of biologics and biosimilars as well, to really understand what the best decision-making tools are available. So several of our clients have submitted data generated using our assays to the regulators, depending on the specific questions that they're being asked. So in total, this sort of approach can really support a quality by design strategy. Now I'm going to be talking about several different assay types, which, if I just go back to this diagram here, are really zooming in on this issue of MHC peptide presentation and T-cell activation. So for a T-cell epitope to be a T-cell epitope, a few things have to happen. First of all, the peptide has to be processed. It then has to bind to the MHC molecule, and a T-cell then has to recognize it and be a functional T-cell. So these three things have to happen, and to do that you can't really ask that with a single assay. You have to do several different approaches. Now many of the in vitro assays that we carry out at ProImmune are using panels of healthy donors. So these healthy donors are typically sourced from the UK population, and these are anonymous altruistic blood donors, so we receive cells from those donors, and we fully HLA type them for DR, DP and DQ. These are pre-stocked, so we process them, we cryopreserve them, and then we pull out those donors for the assays. It's a limited supply of each donor because they're just through standard blood donation, so we can't go back to those same donors again and again. But the key thing here is that we're able to use a broad selection from the population. And one of the nice things about looking at a broad selection from the UK population, for example, is that we have a very broad HLA type. And so not only are we looking at HLA-DR, but we also select our donors on the basis of DP and DQ, which are also very important for the overall CD4 T-cell immune response. From the majority of our assays, we also do a bead depletion step for CD8 because we're only interested particularly in looking at the CD4 T-cell responses. And of course, all these samples come with full consent, and we are a UK Human Tissue Act licensed premises, which is required by law. The other approach that we have for sourcing samples, which is for some of our assays, is actually using fresh blood samples. And for this, we recruit donors specifically for particular studies. This is most relevant for our cytokine storm assays, which I'll be talking about as well. And here, we actually register our donors directly through our website, and we can recruit these donors very quickly. Normally, these are healthy donors, but we can also recruit disease state donors where that's possible. But of course, it depends a little bit on the frequency of those donors. And because we have direct contact with them, we can also invite them to come for further donation if they're willing, so we can do follow-on analysis as well. So the first assay I'm going to talk about is called our Propresenter antigen presentation assay. Now Propresenter is an assay which directly measures MHC peptide presentation on monocyte-derived dendritic cells that have been cultured with a protein of interest, and we use tandem mass spectrometry to sequence the peptides that are naturally processed and presented in the context of MHC molecules. Now this isn't a technology which has never been done before, but one of the key things to bear in mind with Propresenter is that we are the only commercial provider of this technology. Now one of the advantages is that we have really taken advantage of our know-how with regards to cell culture and antigen presentation, and really combined that with the sensitive and increased sensitivity of detecting peptides that are naturally processed and presented using the latest tandem mass spectrometry approaches. So tandem mass spec itself allows you to look at femtogram levels of peptides that are presented in the context of MHC molecules. So this cartoon overview just shows you exactly what we do. So first of all, the antigen, which is typically provided by our clients, is loaded into immature monocyte-derived DCs. The DCs are then matured, and this causes upregulation of MHC class II, and peptides derived from this antigen are then naturally presented in the context of the MHC molecule. We then carry out a lysis step and an immunoprecipitation to pull down, for example, HLA-DR. So we can look at DR, we can look at DP, we can look at DQ individually. Following the immunoprecipitation step, we then recover the peptides, and these are then sequenced through tandem mass spectrometry to identify the individual peptides that are presented. To do this, we need to know the sequence of the antigen, and we also need the full human proteome sequence, of course, in the database. So we actually generate a unique database which contains the full human proteome plus the full sequence of the antigen of interest. There are a number of different kind of QC steps which are needed to carry out this analysis. So what we're really doing is we're able to match the actual protein sequence of the loaded antigen. So we're really identifying which peptides are presented to a high degree of confidence. This is using standard proteomic methodology. So we have a greater than 95% confidence-based exclusion search. We also generate essentially a scrambled decoy library, which allows us to determine the false discovery rate. So the decoy library is the full human proteome plus your protein sequence that has been scrambled. So it's like reverse order. And then we repeat the search with exactly the same criteria. And of course, you wouldn't expect any of those peptides to come up unless it's completely by chance. So it's this chance of less than 1%, which is, again, publication standard for the false discovery rate. So when we actually look at the peptides that are identified in this methodology, we see that the peptides have an expected range of lengths of presentation, which typically is a distribution, but typically is around about 15 amino acids. They can be slightly shorter, slightly longer. And we have a number of different criteria which we have for our quality control. We're obviously looking at the characteristics of the dendritic cells. We also look particularly at the detection of endogenous housekeeping proteins, and these are listed here. So for MHC class II antigen presentation, we have this panel of CLIP, LAMP3, TFLC, FC receptors, and upper lipoprotein B, and ITGAM. So peptides from 3 out of these 6 housekeeping proteins have to be detected in the donor for that donor to be accepted into the assay. So just to explain this in a bit more detail, it will be useful to give a case study. So this case study relates to adalimumab, or Humira, which is an anti-TNF-alpha antibody, and typically used in several autoimmune indications. It's an IgG1 antibody which has human-derived heavy and light chain variable regions with a human IgG1 kappa constant region which is generated by phage display. Now, I think these figures change depending on publications you read and what's said, but essentially, although this is a fully human technology, the immunogenicity of Humira is far in excess of what was originally anticipated because of its fully human nature. And so now we see that at least 20% of patients developing low-titer neutralizing antibodies against Humira. So our aim in this project was to identify and characterize those individual peptides from Humira that are naturally processed and presented in the assay. So we take a subset of donors. Now these donors that we use in this particular assay, we took 20 donors, and they were pulled out of our donor bank on the basis of having a broad HLA distribution. And this is just a sample subset of 4 different donors. So we have donors, the donor identity down here, donor 3, 5, 9 and 10. And this is their HLA restriction in the context of HLA-DR listed here. So donor 3 is DR4 and DR15. Donor 5 is DR1 and 0403. Donor 9 is DRB1 0101, 1302, etc. And in the sequence, this is the actual identity of the peptides from Humira that were identified in these specific donors. So one question I think that some people are surprised at is the number of peptides that are identified, or maybe the relatively low number of peptides that are identified from Humira. Of course, we're identifying a very large number of peptides overall. The majority of those are housekeeping proteins. But from Humira itself, you identify specific sequences that can either be discrete, or they can be from several different regions. So this donor 5, we've identified it from 3 different regions, the light chain and 2 sequences from the heavy chain. And for this particular donor, a very large number of slightly different overlapping sequences. So interestingly, this particular peptide regions, or 78 to 92 or 77 to 94, that particular region was identified in nearly half of all 20 donors that we looked at. So a very promiscuous binding peptide. It's important also in these assays not to just look at a single donor. We've already heard from other speakers about the variability between in immune responses in healthy donors. And in these sorts of assays, it's very important not just to look at a single donor in isolation because of antigen processing differences that there may be on a donor-to-donor basis. So we take a broad view, and typically in these assays, we wouldn't really look at anything less than 10 donors. So when we actually summarize all the data from Humira from this panel of 20 donors, we've identified 4 sequences from Humira. This particular sequence is the one that's really promiscuous and comes up in 10 out of the 20 donors. And if we look at the HLA distribution or the HLA restriction of the donors that this peptide is presented in, we can see that actually there are a relatively small number of HLA molecules, despite the fact these are 10 donors that this peptide has been identified from. Now, if we then map those 4 regions onto the molecule, interestingly, these are typically in the CDR regions of Humira, which isn't too surprising really, but this is very key information because it allows us to identify precisely those peptides that are naturally processed and presented. If that was all the story, that would be it, but of course it never is. And just because a peptide is presented doesn't mean to say that it's a T-cell epitope. It just means that you've loaded a dendritic cell and these peptides are presented by MHC, and after all, that is the job of the dendritic cell to process peptides. So the key thing is, what about the functionality of these peptides? Are they actually important in reality? So we'll come on to that in a moment, but just to give you some idea of how Propresenter has been used by our clients to date, it's quite a broad range actually. I mean, with most of us here are particularly interested in looking at unwanted immunogenicity, and in that context we've identified peptides from adalimumab, infliximab. We've also looked at peptides from allergens, from protein mixtures. We've also looked at quite complex antigens, so not single recombinant proteins, but you can use this in a vaccine discovery approach where we've actually loaded complex bacterial pathogens looking at peptides presented in MHC class 2. We've also looked at class 1, peptides that are presented directly by tumour cells. So rather than using the dendritic cells, we've taken the cells directly from clients, and then we've done the immunoprecipitation on MHC class 1. We've also looked at peptides that have been presented from haptenated proteins loaded into dendritic cells. And this is very interesting because originally I think our hypothesis was very much, well, are the haptenated amino acids presented naturally in the MHC molecule? It turns out that actually the haptenation itself radically altered the repertoire of peptides that were presented by the MHC molecule. So again, it's a hypothesis-driving approach. And we've also worked with it's a publication for Sanofi Pasteur, which was published at the end of last year, looking at the identification of the CMV epitopes as well for class 2. So our peptides that are identified by mass spec in this approach, are they functional T-cell epitopes when we look at healthy donors? So we've heard already from Marissa that the PBMC Tcell assays can be a useful approach to identify specific T-cell epitopes. So at ProImmune, we have a slightly different approach to the way that it was done at Amgen, and the PBMC Tcell assay that we use is based on CFSE, and it's a flow cytometry assay rather than a tritiated thymidine incorporation assay. So this assay is used to functionally confirm specific T-cell epitopes. So here we take a panel of healthy donors, typically 40 or 50 donors depending on the project requirements, and these are all CD8 depleted as before. The donors are selected to represent a broad HLA distribution for DR, DP and DQ, but in any particular project we can skew that if required. Overlapping peptides are then generated for a protein of interest. So if this is done from scratch, we would take your, for example, a variable domain. If it was a novel scaffold protein, you might want to look at the whole thing. Or you might want to look at a junctional region where potentially neoepitopes may have been introduced. Alternatively, we can take specific peptides that have been identified from Propresenter. Then we do a 7-day co-culture of the PBMCs with the synthetic peptides, and we measure proliferation in a CFSE assay. So the simple way to describe this assay is that the overlapping high purity peptides are added into CFSE labelled CD8 depleted PBMCs, and CFSE is an intracellular fluorescent dye, and when the cells divide, equal portions of the dye splits out and is incorporated into the daughter cells. So what we're able to do, if I go on to the next slide, is identify populations of cells where proliferation has occurred significantly. So as I say, it's a flow cytometry assay, and the advantage of that is that you can, of course, co-stain with several markers. And our standard approach is, as well as looking at CFSE, which fluoresces in the FL1 channel, we also look at CD4 as well. So automatically we're gating already on live lymphocytes, and then we're staining for CD4 and looking at CFSE expression. So in our unstimulated controls in this upper right quadrant, these cells are CFSE high and CD4 positive. So these cells have not proliferated. And then natural turnover will occur, which means that then some cells will then fall into this quadrant A, where they're still CFSE positive, but they've become dimmer. But of course they are CD4 positive high. Test articles, or all the individual test peptides, are also incubated in those cells, and you can see here where you have a strong amount of proliferation, then the cells move from quadrant D to quadrant C. Now, for those of you who are very familiar with CFSE, you can sometimes see these individual populations of cells, but what we're looking at here is a 7-day assay, so we're looking at the total cell proliferation after that time point, rather than looking at day 3 and day 4 and day 5, whereas those points you can see the individual generations of cells having proliferated. So we're looking at total proliferation at the end of this time point. And then what we do is we actually analyse each test article, test peptide in sex duplicate. So this is 40 donors, sex duplicate analysis, and then we measure proliferation above background. So we don't use a cell division index, which is something that we used to do. This is now a proliferation that's determined by subtracting the unstimulated control from the test article, test peptide. And that's because with a cell division index, you're dividing by the unstimulated response. And if you have very low cell turnover, you're dividing by essentially 0, so you get a very infinite figure for the cell division index. So we report this now as a subtraction. So we're looking at the proliferation to the peptide minus the proliferation to the unstimulated control. And a significant response is then determined by statistical application of the data and an over-analysis, and we have 2 standard errors of the mean above background. We also are able to report the data with different levels of cutoff as well, and that's something which our standard report would also include. So this is the data where we're looking at the percentage antigenicity on the y-axis and all the different peptides on the x-axis, sorry. And this is looking at Humira variable domain, and all the individual peptides are along the x-axis here. So the percentage antigenicity is the percentage of donors that have responded significantly to a particular peptide. So this red bar means that one donor, which is 2.5% so one donor out of 40, has responded significantly to this peptide 75. The colours are consistent, so each different colour is a different donor. So you can see that we have some donor here, this red donor is quite a hyper-responsive donor. And then you can see that there are some peptides, such as peptide 107 here, where we have 7 donors out of the 40 have responded significantly to that one particular peptide. Now if we overlay the CDR regions for this antibody, you can see again, not too surprisingly, that the majority of the functional T-cell responses overlay with where the CDR regions are of the antibody. Now I want to just bring your attention to peptide 102, which lies just outside the CDR3 region. And this particular peptide is actually the peptide that we've just been identifying and talking about earlier in the Propresenter assay. So we know that this particular peptide, which is very promiscuously detected and presented by a range of different MHC alleles, is also a functional T-cell epitope in healthy donors. Now, of course, ultimately you want to then measure this in patients to see whether this is a functional response. And I think we're probably still at the same point as we were when we were talking about this last year, where that is still, I think, the goal, to identify these particular epitopes in patients. But of course, those things, getting hold of that material is also very, very difficult. But nevertheless, we've identified peptides from Propresenter that are confirmed to have a functional response. These assays have also been deployed in many other areas as well, and this is an example of a publication from last year from DSTL, which is a branch of the UK Ministry of Defence. And this was a project where the client was developing an anti-Venezuelan equine encephalitis virus antibody. Try saying that. And that antibody was used as a passive immunization strategy. The molecule itself, or sorry, the virus itself, Venezuelan encephalitis virus, had been made into a sort of weaponized version, and so having antibodies against it was very, very important as a way of vaccinating troops before they go into the field. And so the client had developed a chimeric antibody against VIV, and also a humanized VIV antibody as well. So the first issue was that the chimeric antibody itself was very difficult to express, and they'd changed the actual sequences by point mutation, and then the humanized version was then developed. Now we generated all the overlapping peptides for these two different variants, and then carried out the analysis in the PBMC Tcell assay. And we can see that actually the peptides that actually continued to respond, these peptides were from the CDR regions, which really couldn't be manipulated very much. But remarkably, you can see that the T-cell antigenicity is significantly reduced with the chimeric, now comparing it to the humanized, so significantly reduced in the humanized version. It's not always quite that cut and dry and that black and white, because the majority of molecules which you screen will have some inherent T-cell responses. But this is quite a dramatic example of where manipulation of the sequences between the chimeric and the humanized version have really reduced that T-cell epitope content. Where we would caution this sort of data is really sort of translating that into what the clinical immune response would be. And this really goes for all of these sorts of in vitro assays, because just because you get a reduced immune response in these assays doesn't mean to say that the clinical immunogenicity is necessarily going to be reduced as well. We would hope so, and it's the best approach to obviously eliminate T-cell epitopes, because the T-cell epitopes are required to drive that immune response. But as we've seen from many of the other presenters today, there are many other factors which of course cause that immune response, notably the HLA restriction of the patients, the route of administration, aggregation of the molecule. There are so many other factors which come into play, whereas here we're just looking at synthetic peptide overlap. So all we can tell from this assay is really the importance of individual T-cell epitopes in that particular protein sequence, which is very valuable information from the point of view of the design of the molecule, but it doesn't tell you everything. So one other question that people would have is, if a peptide is presented in and identified through the Propresenter assay, what is its HLA restriction? Because in the Propresenter assay, we're doing an immunoprecipitation using anti-HLA-DR, for example, and each individual will be expressing two different DR alleles. It might be DR1, it might be DR7, for example. And so therefore you might want to really drill down into more detail and characterize the specific HLA restriction of those peptides that are presented. So to do this, we have developed an HLA binding assay called REVEAL, and this is a high throughput physical assay which doesn't rely on in silico approaches. And in silico approaches do have their value as an early screen potentially, but they still have many inaccuracies associated with them, and I think the field is still quite young and there's a lot to be done still. So one of the nice things about this assay is that it can confirm very quickly which peptides will bind to what MHC molecules. And at Prairie we have a very broad coverage of DR, DP and DQ alleles, in fact more than 56 different alleles to give a greater than 90% global coverage. So this allows us to rapidly identify the specific HLA restriction of individual peptides, and of course you can then use that even at the design stage where you're introducing point mutations to see whether that's going to enhance or reduce binding to a specific HLA allele. The principle of the assay is really, it's an immune assay which involves a conformational antibody that binds to MHC peptide complexes. So we screen individual peptides, typically overlapping, but again they can be specific peptides that have been identified in other assays. The peptide then binds to the recombinant MHC class II molecule, and that binding event causes a conformational change, and then we have an antibody which binds to that whole complex, and that's measured quantitatively in an immunoassay. Now there are two components to the binding assay. We have an initial phase which looks at how much complex binds at time 0, and then we have a stability assay, which is where we actually heat the complexes up to 37 degrees over a 24-hour period, and then we can measure how much peptide remains stably bound after that period of time. And that's really important because with T-cell epitopes, the peptide really has to be engaged with that MHC molecule for a decent amount of time for it to be presented to a T-cell receptor. And that's quite different from, for MHC class 1, the affinity of the peptides is much stronger, whereas with class 2, the interaction is much weaker. So being able to distinguish different peptides in relation to their stability is quite a nice feature of this assay. It's just a list of all the different HLA molecules that we have available, just to point out DPs, DRs and DQs. The field of even putting this list together is quite complex. I shan't bore you with all the details, but the alpha and beta chain pairings for DQ is really still at the cutting edge of our knowledge. And when you do an HLA type of a patient, you'll only now just begin to get both the alpha and the beta chain information from those individuals. So that's quite useful because it allows us to then put together this panel of alleles which are the most common chain pairings of both alpha and beta chains for DQ. With DR, you're always talking about the same alpha chain and also DP, but for DQ there was a lot of heterogeneity. So back to the Humira story. This is our peptide 102, which we identified in both the Propresenter assay but also in the functional PBMC Tcell assay. So I'm just going to zoom in on this particular peptide as a way to describe the story. So this peptide, amongst others, was screened in the binding assay, and these 5 donors here are actually 5 donors that responded significantly to this peptide in the PBMC Tcell assay. And if we look at the HLA restriction of these donors in the PBMC Tcell assay, we see that they're DR1301, 1501, DQ0602, 0603, DP04 and DP02, etc., etc. Now the colour codes, the traffic lights, relate to the strength of binding of this particular peptide as determined in the binding assay and stability assay against those specific alleles. So we actually screened a very large panel of alleles, but I'm just zooming in here on the functional responses that we were measuring in the donors. And we can see from this that actually the strongest binding was observed in DR0404, 0101 and 0301. So red is no binding, grey is not tested because that allele wasn't in our panel, and yellow is weaker affinity binding. Now I'm just going to, it's interestingly enough, this particular peptide is just clearly DR restricted and probably not DQ and DP restricted. Some peptides we see there are in relative importances of DQ and DP. It's not always the case that DR is always important, sometimes it isn't. But I'm just going to hide them for the moment because in the Propresent antigen presentation assay we were using a DR immunoprecipitation. So I'm just going to focus on DR just to keep the data a bit simpler. If we come back to this data here from the Propresent assay, we can see that this particular peptide was identified in a DR1 restricted donor as well as a DR3 restricted donor, and all these other alleles as well, which we don't know whether that peptide was restricted to, but that's what the donors were positive for. And here, now combining this with the binding assay, we can tell that actually, for certainty, that this particular peptide, which is promiscuous, is certainly restricted to DR1 and DR3 by looking at the reveal binding data. So what do we do with this information? Well, on one level, this has really zoomed into the nth degree, because we now know that this particular peptide from Humira is restricted to DR1 and DR3. That does mean, at least in patients that are receiving Humira that are DR1 and DR3, we have a potential biomarker to then track those immune responses to. So we can take a blood sample from those patients, we can stimulate with a single peptide, we can then measure the immune response, for example, one way of looking at it in ELISpot or flow cytometry assays. The issue, of course, with measuring patient material is that you're always limited by the amount of cells that you can get. So anything which can zoom in on a single peptide here, you'd have to obviously add in all the relevant controls, but at least you're talking about 10 mils of blood, something that you can work with, rather than to have to do a whole panel of screening, which would mean several hundred mils of blood and something that wouldn't be feasible. So this level of detail means that you can really zoom in on a specific sequence from your biotherapeutic and demonstrate to the regulators that you've really done your homework and these immune responses can be tracked in particular patients. I'm not going to spend too much time on this because actually David was showing the data earlier, but this is a case study from Pasteur where we actually used our binding assays to characterize binding of a panel of different factor VIII derived peptides in a number of different, or 7 different HLA-DR alleles. And so here we just see that the combined data set that allows us to look at the promiscuity of binding of these different peptides to different degrees in these different sort of sets of peptides. And this information was used to really understand in their DR2 transgenic mouse model the importance of these different sequences. Okay, the next assay I'm going to talk about is our dendritic cell T-cell assay. Now this is something which is very sort of popular at the moment, particularly because it allows you to sort of stand back. And I think the problem maybe conceptually people have with some of the other assays is that it's really looking at a lot of detail. You might not be at that stage, you might not really care too much about the individual T-cell epitopes. You're at that stage where you've got lead X, Y and Z, and you want to make a choice with regards to which ones to take forward, all other parameters being equal with regards to efficacy, PK/PD, potency, but you don't know about total T-cell epitope content. And this is where the dendritic cell T-cell assays come into their own. So similarly to the other PBMC assays, we take a panel of between 20 and 50 donors. Our sort of standard is 40, so we 20, 40 or 50. Again, this typically represents a broad HLA distribution. We generate dendritic cells from monocytes in culture, and then we load with the protein antigen of interest. So these proteins are provided by our clients. They may be novel leads, they may be really comparing existing marketed drugs to novel leads. They may be biosimilar versus innovator molecule. So there can be lots of ways that you can really design this assay. The key output is that it allows you to determine the relative antigenicity of different drug candidates directly. So this doesn't tell you, again, exactly what's going to happen in the clinic, but it does tell you about T-cell epitope content, having taken into consideration natural processing and presentation from the protein of interest. So the proteins are loaded into the immature DCs, the DCs are matured, and at that point then peptides are presented from the protein of interest in the context of MHC class 2 to autologous CFSE labelled PBMCs. So these PBMCs are derived from the same donors that the DCs are, so there's no unwanted interaction there. And then we measure proliferation just as before. Again, high numbers of replicates are incorporated into this assay, and it's a relatively rapid turnaround. So actually we've worked hard to reduce this, and so for a 40 donor study with a panel of sort of 6-7 different proteins, for example, we can turn that around in about 5 to 6 weeks. The maximum number of proteins that we can measure simultaneously is obviously limited by the numbers of cells that are available for each donor. I think the most that we can accommodate, including the control antigens that we have as standard assay controls, is about 18. It may be possible to do a few more, but it's that kind of number. So the output of this assay we prefer is a response index, and the response index takes into account the proportion or the percentage of donors that respond significantly to a particular antigen, but also takes into account the strength of the response of the T cells. And that's important because with dendritic cells you've really got the advantage of a potent kick to the T cell response. So not only do we look to see whether a donor responds significantly, but we also see what the strength of that response is as well. So a response index is the percentage of donors responding multiplied by the average strength of response. And this data here demonstrates an example of several different molecules which are undisclosed here, but we have a chimeric antibody, humanized 1, 2, 3, 4, 5, 6, and a fully human antibody at the end. And these are our control antigens, which actually are off the scale. This is KLH, keyhole limpet hemocyanin, which is an internal assay control for naïve T-cell responses, and tuberculin PPD, which serves as a strong memory T-cell response. And in the UK, because BCG vaccine is only now beginning to sort of phase out, all of our donors have very strong PPD responses, so that's quite a nice control. If you're thinking about doing these assays, it's very important, of course, to think about the design of the molecules that you're going to be comparing. It is a comparative assay, so the more donors that you have in the assay, the more similar the outcome will be when you repeat that assay, exactly the same molecules, but on a different set of 50 donors, for example. The key thing, though, is to think about that this is a comparative assay, so you can design it to make sure that you've got your different leads that you're trying to compare, plus, for example, an innovator molecule or the marketed drug, so that you've really got the right comparisons in the assay from the outset. So by integrating different assays, depending on the stage that you're at, this enables, or has enabled us to look at the antigenic immune response to Humira and the characterization of it in a very short amount of time, for example, just 8 weeks. And this can be applied, as I've mentioned earlier, in different phases, whether you're thinking about selecting different molecules, so a QBD strategy for lead selection. It could be that you want to identify epitopes to then monitor immune responses in patients downstream. It may be that you're thinking about providing a more detailed data submission to regulatory authorities, particularly for novel proteins. We've heard about X10, for example, or actual other therapeutic proteins directly. So there's lots of different ways that this can be applied. Okay, so the next... Oh, I'm just realised my time's up. I shall be very, very quick. Just carry on. So the next thing I was going to talk about is ProStorm, which is our cytokine storm assay. This is a first infusion. This is an assay which allows us to assess first infusion reactions that occur in response to biotherapeutics. It's a slightly different area. The issue of cytokine storm is obviously quite a challenge, particularly relating to rapid production and upregulation of cytokines in response to factors which are quite complicated, but different molecules following administration that can occur within minutes. Now we all know about the T. Genero drug 1412, which caused some very, very serious adverse effects in all the test subjects that received this molecule in London. And the way that we want to address this is actually by using a very simple whole blood assay. And in this assay, we work with a panel of donors, for example, 30 donors. Fresh blood is drawn into anticoagulant. We then add a range of different test articles in a range of doses, and we have controls for the assay as well. We also include a low response comparator and a high response drug comparator, and then we're able to incubate those molecules in a very short assay time. So literally you're taking fresh blood from fresh donors, adding the drugs into culture pretty much within a matter of minutes, and then going on and doing the analysis. And the analysis the analysis we look at is looking at plasma levels of a panel of different cytokines, of which a small proportion of these are actually asked for by the FDA, but we do a much more extensive panel using our Prairie Ultra platform. And the nice thing is here, if we look at IL-6, just as one example, just to take you through the data, these diamonds denote different donors. So each donor is a diamond, and when it reaches a threshold, which is set, it's an arbitrary threshold set on our low response comparator, Erbitux, which is 95% of donors are going to be negative for this. We can see that as you increase the dose, then more donors switch over to the right-hand side. So at this point, this is looking at Remicade. We can see that as you increase the dose from 0.1 up to 100 micrograms per mil, then 1, 2, 3, 4, 5, 6 donors out of the full 29 are significantly responding here for IL-6. This is a logarithmic scale of the cytokine concentration in picograms per mil. We were talking about aggregates earlier. This is, again, just looking at IL-6. But what we can see here is Remicade, aggregated Remicade. This was stir-stressed overnight. Erbitux, Campath, Herceptin, and Vectibix. And the picture is, as you can see, that as you increase the concentration of aggregated Remicade, suddenly you get this threshold at 10 micrograms per mil, where all the donors are suddenly lighting up. And it's this aggregation effect which is causing a strong response and production of IL-6 into the whole blood assay, which is then measured in this multiplexed system that we use. You can see other molecules like Campath, which is a positive control essentially, and Erbitux, which is our low response comparator. So this information can be used as a way of understanding relative risk of different molecules with regards to production of a range of different cytokines. So you can get a feel for where your molecule lies on a scale. We use a system called Prairie Ultra, so I'm just going to make this my last slide. Unfortunately, I haven't got time to talk about other things, but I'll be very happy to discuss it later. But Prairie Ultra is a tool which is a high-content protein peptide microarray. We use it for B-cell epitope mapping. We can use it for anti-drug antibody mapping. Essentially, it's a very high-content array where we can print more than 30,000 analytes on a single chip. And we also use this to capture antibodies onto the array. So, for example, anti-IL-6 is then layered with the plasma sample, which is then layered with an anti-IL-6 detection antibody. And we can use this. The key advantage is that we can work with very small quantities of material, literally just a microliter. And there are publications for this as well now, recently looking at B-cell epitopes from OSPC protein in Borrelia burgdorferi sensu stricto, which I have never had to say before. So this platform is sort of integral to how we detect the cytokines in our ProStorm assay, but we also use it to do B-cell mapping as well. So I won't go into this in too much detail, but just to say finally that the toolkit that we have available allows different questions to be addressed at different time points. So whether you're thinking about natural antigen processing and whether that is an important thing to consider in your assays, then in that case the DCT Selassie and the Propresenter assay both take into account that aspect. If you want to look at physical peptide binding to the MHC, then obviously we have the binding assays, and we can also confirm that in the antigen presentation assays as well. T-cell functionality is taken care of in the T-cell assay and the DCT Selassie, and actually identifying epitopes through the binding assay, the T-cell assay, and the antigen presentation assay. And then ProStorm is sort of out on the limb there at the end. But all these different assays sort of integrate depending on the kind of questions that you have. And we're always looking to innovate for new sorts of assays as well. So if anybody has any particular gaps in their knowledge, then I think sometimes it's a great idea to sort of discuss some of these things, and hopefully in our forum tomorrow, or even in our discussion now, there'll be an opportunity to bring up some of those points. So I'd just like to say thank you very much for your attention, and I'll be welcome any questions. Yes, Judy.
Did you check whether TGN 1412 scores in your ProStorm?
We would love to. We would love to. We can't get hold of it.
No?
Yeah. I don't know if you have any comments on that, Nick, but yeah, we weren't able to get hold of any 1412. But obviously it'd be very, I mean, there are many different ways these assays can also be sort of set up, either with whole blood assays or frozen PBMCs, fresh PBMCs. And we've decided that the whole blood assay is very important because it gives a very it gives an approach that doesn't affect, or doesn't, what's the word I'm looking for? It's a very non-invasive assay. It's really just taking whole blood from patients and then adding it in. But we would love to look at TGN 1412, but it's just something we couldn't get hold of in the assay, in the setup we did.
Well, yeah, I mean, possibly we could get a hold of it, but we decided not to develop the assay around it because we couldn't get a hold of it consistently in the future as assay control as a formulated drug, because it's now owned by another organization that's taken it on, and I'm not sure they're really producing clinical batches. Roche have worked with the same assay and we consistently get a positive response on that antibody. So Roche have worked on this type of assay, and they've used CD28 super agonist, and they do consistently get positive. So you could buy research grade CD28 antibodies, and you would probably find that you get 100% positive result in this type of assay. But it's not a clinically formulated drug, so we don't use it as an assay control. And Campad is a very good positivity assay. It comes out positively every time we do it.
David.
So Jeremy, can I just talk to you about the Humira study and ask whether you can exclude the possibility that your peptide isn't binding to MHC and isn't stimulating T cells as a cryptic epitope?
I think you probably can't. I mean, the antigen presentation assay, all you can do is see peptides that are there. You're not able to see peptides that aren't. And obviously any assay is going to have a limit of detection anyway. With regards to the overlapping strategy, you're generating synthetic overlapping peptides. So if you're going down that road, then that's one approach. So I don't think you can eliminate that. What this does allow you to do, of course, is at least identify some peptides that are naturally processed and are being able to identify through that. But I think if something's not there, or at least is there in a way that we're not expecting, then that's not going to, not going to see it in that assay.
Yeah, but I think we have to be cautious before you say, right, I've identified that epitope now, and that is the correct epitope.
Yeah.
And we're going to use that as a biomarker.
No, exactly. No,
That's a stretch.
it is, and you need to go down the whole road of really validating that in a clinical scenario, and we're not there yet. What I'm saying is potentially that's conceptually that's how you could apply it. I'm not aware of anybody who's doing that yet, but that's something which you could potentially do. There would be obviously a lot of work that would have to be done before you get to that stage. Yeah. Yeah, Marissa?
First of all, very nice presentation. Can you hear me?
Not yet.
Oh, just push it once, I don't hold it.
Should go green. Yeah. Yeah, that's it. It's working.
Okay. So I had two questions. The first question was on your DCT cell assay. I was wondering, because the DCs are, you said that you mature them. I was wondering if you see how molecules with low rates of clinical immunogenicity behave in that assay, and if you see an over-presentation of molecules. So that was my first question. If you see like a molecule like Xolair or something that has a very low rate, is that over-presented in that assay? And my other question was regarding the cytokine storm assay. You have some really good controls built into this assay. And I was wondering, how do you then determine if you have cytokine storm? What's the level you're trying to compare to that? Is it just to the CAMPAT level, or is it know about that?
Good questions. So first of all, with the DCT Selassie assay, you were talking about the levels of presentation of that drug. Well, what we're not looking at, we're looking at the amount of class 2 that's expressed, but we're not looking at necessarily, we haven't measured that in the context of comparing it to different drugs. And the question, I think, relating to different levels of clinical immunogenicity, it does come down to the fact of what this assay, in our opinion, does is tell you about T cell epitope content and not about what that clinical immunogenicity correlation is. And that's very difficult because, as we've just been sort of slightly disagreeing earlier, it's very difficult to actually know what that data really is and what those values are. So for us, it's a decision tool to be used along the road in understanding which molecules to take forward on the basis of having a relatively lower T cell epitope content. I don't know if that 100% answers your question relating to DCT Selassie.
Well, I can answer it a bit more. So Xolair comes out low in the assay, and Remicade comes out consistently high, as an example. And it is a relative ranking assay, so we're not saying you never see no T cell response from a low immunogenicity antibody, but it's a comparative assay, and in general terms, it seems to also correlate with what the longer term clinical experience has been with some of these molecules. In terms of the cytokine storm control question, I mean, this is also the question how you really control this assay. And we followed a little bit the lead that Roche have taken on this, that sort of Erbitux is something that they've kind of looked at as a sort of negative baseline. There's of course this hypersensitivity response to it, but failing that, you typically see a very low cytokine response to Erbitux. And CAMPA gives you a very reliably nearly 100% response across the board, so it's a nice sort of positive level. And both drugs are obviously available off the shelf, so that's why we like to use them, because we know we'll be able to buy them in the future and put them in the assay. But yeah, I think every sponsor has to probably look a little bit individually how they like to sort of set their cut points in the assay, but that's just a sort of suggested method, and then you can sort of rank your molecules.
Yeah, so it's still only arbitrary with regards to the threshold. So even the 95% of donors being negative for Erbitux, because you can have pre-existing antibodies to a sugar in Erbitux, which is where you get those sort of single donors coming up positive. So I think there's no perfect control, as we know in any assay, but it's an indication. You do have to think about it based on the molecules you're actually comparing. Okay, yeah, I think Bill has the last question before we move on. Yes, Bonnie.
With regard to the aggregation, did you say how you prepared those aggregates, and also
They
I was
weren't.
wondering if you had any speculation about what the mechanism is there?
Yeah, well, I think mechanistically the speculation is probably to do with really binding to TLR agonists, things like that. It's very much speculative, but we weren't able to characterize the aggregates. We didn't have that ability, but certainly the stir stressing, we know from a point of view of visual inspection, gave a very cloudy appearance to the protein. And it was a bit of a last-minute addition to the experiment, but certainly very interesting with regards to the outcome. And it's something which I think Nick's going to be speaking a little bit more about tomorrow.
Yeah, so it was a last-minute decision to say, oh, can we just stir this overnight before we put it in the assay? So that's what we did at 700 RPM, I think, for 24 hours. And I think we over-egged it a bit. It was all crashed out, and in fact it was a bit difficult to measure the protein concentration that was left. So the values there may not be entirely accurate, so it was a bit sort of a blind addition to the assay. But clearly you see a sort of thresholding at some point as you add more. You see a real kickoff, and you need to remember that this is a logarithmic scale, so the cytokine response is really 100-fold or more